Welcome back. I didn't highlight them, so I'll underline them today. We're going to go ahead with our response to the antigen. So quick, you should know what's an antigen. I want you to be able to tell me B cell versus T cell. I think you should look back at how a phagocytic cell can become an antigen presenting cell, or an APC, and then let's put it all together. I first ask you to identify which of these statements was true about B cells. It's 1 and 2 here. They have Y-shaped proteins, which you can see are not particularly obvious with a microscope. They're very tiny, but these are what allow them and their variable regions to target invaders that are in your humoral, or your body's response. And then these Y-shaped proteins break off and become antibodies. These other two, what kind of cells are these? I think you should come to review. Have to find out. So let's go where we start out. Here is a phagocytic cell. This eats invaders, and it happened to eat this bacteria. This is a pathogen, and this pathogen on it has proteins that have epitopes. This protein is an antigen on a pathogen with an epitope. This phagocytic cell eats it. Nom, 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 nom. And then it displays, I just ate this. Now look, it's just a piece of this. It's just the protein. So now this antigen, that's antibody generator, presenting cell, or our APC, is doing its thing. It's presenting an antigen, and it's a cell. This presentation is on a protein away from the cell's body, and sometimes a T cell, and this is a special kind of T cell called a helper. Its job is to help the immune system. A helper T cell comes and it binds with this antigen on the antigen presenting cell. If they match and they lock together, so the antigen receptor on the T cell binds with the antigen that is being presented, I know this is a lot, they lock together. When they lock together right here, and I'm leaving out some words here, so if you've learned this before, I'm trying to leave out some of the names of the proteins. But as they bind together, the actual binding causes the T cell to respond. It changes what it does. And what it does is it releases cytokines. We've heard cytokines before. Any memory of cytokines? Just from the last lecture. These are paracrine localized signaling molecules that stimulate the immune system. One of the things they're going to do, these cytokines are like yelling for help, 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 help. These cytokines are like yelling for help and saying, hey, there's an invader. I've figured it out, but I need help. These cytokines can summon another kind of T cell. This is called a cytotoxic, or a cell-killing T cell. And it can also summon B cells. When this helper T cell binds with the presented antigen on the antigen-presenting cell, what happens is the cytokines signal that there is an invader, and we need to call for support, because now we're fighting. There's an infection, there's a virus coming, cells are going wrong. We need to fight them off and be healthy. I always imagine cytokines as being like an alarm, like a giant fire alarm going off. So in my town, when there's a fire in town, there's a siren that goes off in the whole town. I imagine that's cytokines. One person notices the fire, they recognize it, they're like, oh, that's bad. They pull the alarm, and now everybody, the firemen, everybody in town, the ambulance, everybody starts coming in to provide help. these helper T cells, not calling the firemen, but they're actually going to trigger two separate responses. We need to destroy cells that are already sick, and we need to destroy anything that has not gotten in yet that's still floating around in the blood. So helper T cells trigger cell-mediated responses. These are those cytotoxic T cells. These kill your infected cells. And helper T cells stimulate the humoral or the B cell response. It says it there, I'm writing it again. In the body's humors, in your blood system and your lymph, where you're fighting with antibodies to kill things in your fluids. That sounds goopy. I guess it is goopy. It kills things in your fluids, but not your infective cells. We want to get rid of it. We don't want it to be able to hide inside of your cells. Calling on all the helpers with cytokines or the alarms. So, we have your phagocytic cell. Nom, nom, nom. It ate a pathogen that was infecting you. Like all cells, most of your cells, it shows a display of what it's been doing. And this cell has been eating invaders. So it becomes an antigen presenter. It presents what it just ate, and it shows it to the world. A T cell, a helper T cell, binds with the presentation of the antigen. They lock together, and this T cell starts releasing cytokines. This is local signals, paracrine, but this T cell is also stimulating itself. It is autocrine talking to itself. What happens is this T cell releases signals that cause it to make more. It calls cytotoxic or cell killer, cell immunity, cell mediated immune system response. It also calls humoral response, the B cells, where we're going to generate antibodies and we're going to signal for the production of killing things that are floating around inside your humors, inside your blood, inside your lymphatic system, but have not yet gotten inside of your cells. So we're just going to kill everything that is infected and everything that's trying to infect, hopefully. The cytotoxic T cells, the cell killer cells, are going to go through this cell-mediated response. This means your cells are going to be evaluated for being infected, and if they are, they're going to be killed. That's okay because you're multicellular and you don't need cells that are killing you. You want to have cells that are working together with the rest of your cells. Now, there's a couple ways that can happen. These cytotoxic T cells start binding with cells that are presenting bad proteins. So this cell is infected, he's sick. This cell is sick and infected, and he's presenting a protein that does not belong inside your body, not you. The cytotoxic T cell binds with it, and binding with it triggers it to react and lock on, and the cytotoxic T cells releases perforin. This is one of my favorite proteins. That's exactly what it says. It makes pores in the sick cell. Well, it makes pores big enough that everything can fall out. And so the insides kind of leak out. And granzymes, which tell this cell to initiate apoptosis or cell death. And everyone always tells me, and when we do this live, everybody's like, oh, no, this cell is, imagine like a zombie cell. It looks like it's you, but it's doing stuff to hurt you, to make you sick, or to kill you. You can't make it better. The only way you can do is get rid of it and make sure it stops making you sick, because it's making proteins, it's making viruses that are actually making you sicker. So you need to get rid of it. B cells do not just have to rely on responding to the entire process we talked about. They can initiate it on their own. So a B cell can respond to an immune response that was triggered by a phagocytic cell displaying, becoming an antigen presenting cell, and binding with a helper T cell. Or a B cell can encounter that antigen on its own. Here in this picture, the B cell has encountered an antigen. That's this word, antigen, or an antibody generator. And because the B cell happened to have the right shape to bind to the antibody generator, it created a successful attachment. It bound. This activates the B cell. The B cell, in response to this, proliferates. It makes lots of copies. Those copies make lots of Y-shaped antibodies. It also displays what it is doing because cells need to talk to other cells in a multicellular body. And when it displays, it can also bind to a helper T cell. And you're like, why? We're already doing this. Sure, helper T cells can call B cells. But who else do helper T cells call? Because we don't know if this is just in the humoral, the blood, and the lymph, or if it's already infected your cells, and we want to get everybody involved. The B cells multiply, they secrete antibodies. The antibodies mark and bind with the target pathogens for destruction. Now, antibodies, I want to show you as we go on through the rest of this lecture, antibodies do not bind with the antigen and suddenly everybody explodes. I don't know why. I always sort of imagined it like pew, 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 like Star Wars. What they do is they bind, and then these pathogens are unable to do much. Let's look at that. These antibodies are part of the complement system, or the system of proteins that complements the antibodies. Now, there's two big things we can do here. The antibodies can neutralize a virus. If they bind to the epitope on the antigen of the pathogen when it's a virus, this virus, it can't enter the cell anymore. Now it's just off. It's broken. We've prevented it from binding to anything. We say it has been neutralized. If it's a bacteria that is infecting you and it is able to reproduce on its own, maybe it's synthesizing something that's making you sick, something that's toxic to you, we're not neutralizing it is not going to be an option. It's not going to be brought into your cells anyway. We can opsonization, it's a hard word to say, opsonize or opsonization, bind to the surface of the bacteria, and then they get so bound up, they get big. Here we got more, and they start to get lumpy and clumpy, and this actually makes them a target of phagocytosis, or somebody's going to come along, a different cell, and eat them. Either way, we remove them from being able to attack your body. The complement system also has complementary proteins. I like these guys. These are my favorites. I know I said that in the last lecture too. I have a lot of favorites here. Complement system can bind to proteins here. It can create pores where the proteins bind, and they are amphipathic integral proteins that scoosh their way into the cell's membrane, and then they open up like a stent in the heart. If you've ever seen a stent put into like an artery in the heart, it opens it up and it creates a hole that stuff can flow through. Except now all the stuff inside the cell is going to flow out. With that lovely picture, I'm going to leave you and we're going to talk about how we stimulate the adaptive immune responses artificially, or how vaccines work. Put them in order. You need to come to review. Remember, there is not going to be a workshop on this. There are not going to be an overly large number of questions on the exam about immunity. 8 on adaptive immunity, 5 or 6 on innate immunity because blood was part of that lecture. But so each outcome will be covered once. But this is tricky, and it requires you to remember a lot of the material we've covered in 116 and 115. So let's sequence it and review, and then we'll see if everyone can get it like awesome correct on the final exam. And I'll see you in the penultimate video, the topic 27, vaccines.